to the research seminar series, the latest in public health research. I see you all connecting to audio, so I'm stalling a little bit as you all get in here and able to hear. I am very pleased to introduce our speaker today, Dr. Bill Jagust, professor in public health as well as in neuroscience. And his work concerns changes in the brain that co-occur with aging as well as Alzheimer's disease. And I believe he'll be telling us about his work on Alzheimer's today. So we so much appreciate your time, Bill, and I'll hand it over to you.
Thanks, Jen. Let me get my screen shared here. And good. I hope someone will tell me if that's not working. So I am indeed, I have a joint appointment in public health and neuroscience, and I walk a funny pathway with a foot in the world of sort of human and clinical research, but also very interested in neurobiological mechanisms. And I'm going to try to give you a big picture, partly definitely about what I've been doing, but also about the field in general, because I know there's a lot of interest. So let me start out with a kind of just an interesting observation on NIH funding. And for a long time, the Alzheimer's disease budget was around $500 million a year. 300, gradually got up to about 500. That's about a tenth of what the cancer budget was. But beginning in 2015, there was really a remarkable transition. And as you can see, the budget for Alzheimer's disease really went up sixfold in about 7 years, to the point where it's gone from 500 million to 3.5 billion. I hate to be so crass as to talk about money on my first slide, but it's really made a huge difference. And it's changed the Alzheimer's disease field enormously. And as you'll see, the work that I'm going to show you all took place in the last 5 or 6 years, in fact. And I think there's been a really dramatic acceleration and transformation of the field for a number of reasons. So let's start out with some very basic definitions. What is Alzheimer's disease? It's the most common cause of dementia. What's dementia? It's a progressive loss of cognitive ability that interferes with daily life. And Alzheimer's disease usually begins with loss of what we call episodic memory, which is memory for the events of a person's life. People sometimes get confused about this because people with Alzheimer's disease can remember things that happened a long time ago. But they can't form new memories. So they can't remember what happened yesterday or sometimes even a few minutes before. And that's, of course, very disabling. And then as Alzheimer's disease progresses, the cognitive loss spreads to all kinds of other areas like language function, ability to make decisions and judgments. And of course, eventually people become dependent on others for their daily life. Now, just one other term to define is mild cognitive impairment, which usually is the very beginning stage of Alzheimer's disease, often memory loss without other cognitive loss. And it's interesting and important because it is frequently a precursor to Alzheimer's disease and very important in identifying people who might be in very early stages of Alzheimer's disease. So there's really a lot of difficulty in doing epidemiological studies of Alzheimer's because until very recently, there's been no laboratory test that is helpful. So it's really been a clinical diagnosis, difficult to make in the field. And epidemiologic prevalence estimates have varied quite a bit, but I picked these because they were, I thought, reasonably demonstrative of a fairly conservative estimate that allows you to see the age-associated prevalence, which really goes up dramatically between the ages of 60 and 80. And in fact, above the age of 85, the idea that 1/3 of people may actually have Alzheimer's disease isn't really a big stretch. It's really within the range of estimates that are out there. So it's a very strong age association and a very common disease amongst older people. you can't understand Alzheimer's disease without understanding what happens in the brain of people with Alzheimer's disease. And I'm going to spend a lot of time talking about the pathological proteins that aggregate in the brain. And there are two proteins. One of them is called beta amyloid, or A-beta. This forms the plaque that you've probably heard about. It's interstitial, meaning that it occurs in the spaces between nerve cells. And it's a sticky, large protein that just aggregates and forms these plaques. The other protein in Alzheimer's disease is called tau. Those occur in the neurons, in the nerve cells, and they form a structure called neurofibrillary tangles. So that's a cellular type of protein. And this tau protein and amyloid protein have been known to be associated with dementia and cognitive impairment for about 100 years, and since Alzheimer actually described them in the early 1900s. What's interesting about them is they're very different in where they occur in the brain and as we've learned in sort of what they do. So the tau protein are very, very common in aging and occur particularly in this part of the brain called the medial temporal lobe. If you don't know neuroanatomy, don't worry about it. I'm going to show you some images, but you'll get a handle. You'll get a feel for it. This is a part of the brain that's very much involved in the memory system. So again, very consistent with the symptoms of Alzheimer's disease. These proteins, this tau protein, accumulates in the medial temporal lobe memory system as we get older. The amyloid protein, in contrast, appears diffusely throughout the brain. And what I want to show you is how common they are with aging, especially the tau protein here in purple, the neurofibrillary tangles, which are ubiquitous. And by the time a person gets over the age of 70, the chances are pretty good that they have tau in their brain. So very, very, very common. And again, this tau is very frequently found in this medial temporal lobe region in the brain. So let's just talk about some of the factors associated with Alzheimer's disease. And a big one is genetics. The genetics of Alzheimer's disease is actually quite complicated, but there's a simple way to understand it. And that is, this is a great way to demonstrate it, where what you see on the x-axis is how common the gene variant or mutation is in a population. And what you see on the y-axis is what kind of risk it confers. And what you can see right away is there's a cluster of very rare, but very, very high-risk genes that are inherited in an autosomal dominant fashion. These are all genes I'm going to tell you about in a minute. But these genes, if you have a mutation in these genes, these are causal. These cause Alzheimer's disease. Fortunately, for populations, they're very rare, but they've been very important in our understanding of Alzheimer's disease. The next most common and important genes is this gene called apolipoprotein E. And if you have the E4 variant of this particular gene, it's a polymorphism, it's just a variant. If you have two copies of this E4 allele, you're at fairly high risk of Alzheimer's disease, at least tenfold over someone who doesn't have the gene. If you have one copy of the allele, it's more common to have one copy, but the risk isn't quite as great. So these are the big genes that are associated with Alzheimer's disease. And then there's a whole cluster of other genes that are either extremely uncommon or have very, very small effects. And these genes I'm not going to talk about at all. They're very interesting because we think they're important in polygenic, in sort of a polygenic risk for Alzheimer's disease. They likely interact with one another. And in the aggregate, they may be responsible for many cases of Alzheimer's disease. But they're very complex in terms of their interactions, and it's very difficult to understand exactly what's going on in terms of their interactions. Okay, so one of the things that's really kind of really changed our field is the availability of biomarkers that we can study in humans during life. So the first one that came along was a biomarker for beta amyloid, and actually we could measure this in spinal fluid for a long time, but it was really PET scanning with a number of different radioactive molecules that sort of enabled this field to just blossom. And one of the PET scanning molecules I'm going to talk about is a compound called PIB. If you see this PIB, you'll just know that we're using it to measure brain beta amyloid. within the last 5 years or so, there have been a number of really interesting papers that have shown we can also measure this beta amyloid in plasma, but we're not quite there yet as far as standardization and scaling this up. So amyloid PET scanning came along in the early 2000s, and tau PET scanning came along in around 2012 or 14 or so. We had this also in CSF for a while, but again, it was really PET imaging that changed things dramatically because we could actually visualize exactly where the tau was in the brain. And although these were available in CSF, it wasn't clear that they were accurate until we had PET scans to sort of compare them to. And most people and most researchers would rather use PET scanning than doing repeated spinal taps to study this disease. So the CSF really never took off. And again, we can now measure tau in the plasma as well. And then finally, there's a whole host of biomarkers that measure a process that we sort of lump together as neurodegeneration. And this mainly reflects brain atrophy. That's mostly what I'm going to be talking about. This is an MRI scan. And in Alzheimer's disease, the brain shrinks in characteristic locations. And this is, while not diagnostic of Alzheimer's disease, it's a good indication of sort of the effect of Alzheimer's disease, the effect of these abnormal pathological proteins. And it gives us an idea of how a person has progressed and how severely affected they might be by Alzheimer's disease. So I want to talk for a couple of minutes about risk factors, and I'm going to come back to this when we get into treatment. There's really a very large literature looking at risk for Alzheimer's disease. Initially, there were case control studies. More recently, there have been very large longitudinal cohort studies. And not every risk factor is validated, but I think the ones that are clearly validated are age, family history, the genetic risks that I talked about, and then this group of risk factors that comes out repeatedly in studies, and I really sort of lump together as evidence of clear vascular disease or risk factors for vascular disease. So the risk factors include things like hypertension, hyperlipidemia, obesity, evidence of heart attack and stroke, diabetes. And then, so these come out very, very clearly in multiple studies. And then many studies have also suggested that depression, a past history of head injury, and a history of sleep disturbance has also been associated with an increased risk for Alzheimer's disease. And then there are interesting constellations of protective factors, which I think also group together in a way that I think might be quite obvious to you. There's greater physical activity seems to be associated with a lower risk of Alzheimer's disease, more lifelong cognitive engagement that can be measured in all sorts of different ways, dietary factors, higher education, and higher socioeconomic status, and as well as higher parental education and higher parental socioeconomic status have all been associated with a lower risk of Alzheimer's. Now, I'm just going to show you a couple of slides that sort of follow up on this protection story, because again, I'm going to come back to it later. And this is some work we did a while ago, showing that actually you could look at lifetime cognitive activity. This is quite a biased measure. It's a retrospective scale where we ask people to rate what they did sort of in terms of their cognitive activity, things like playing games, reading, going to the movies, going to museums, et cetera, looked at across the lifespan in relationship to how much amyloid was in the brain. This is PIB. This is the PIB scan again. And you can see that on average, the higher ratings for lifetime cognitive activity, the less of this amyloid protein in the brain. And this is just a model. This is modeled data to show you that in an individual who was abnormal with this or who had this ApoE4 allele, if they had low cognitive activity compared to high cognitive activity, they would have visibly more amyloid in the brain. And I'm showing you this because this has been another trend in the literature that has been sort of difficult to see, but there are a number of papers supporting this kind of idea. And here's a paper that just recently came out where people were followed during life their early lifetime cognitive engagement was, or cognitive enrichment was measured again with a retrospective scale. So that's biased, you have to keep that in mind. But these individuals were followed during their life, and at post-mortem, their Alzheimer's disease pathology was measured. And you can see that the greater early life cognitive enrichment they had, again, similar measures like playing games, going to museums, things like that, the greater cognitive enrichment, the less Alzheimer's disease pathology they had. So this is a really interesting kind of finding for reasons that are probably totally obvious to you. And again, I'll come back to this a little later in the talk. So I'm afraid I have to talk a little bit about the molecular biology of this, but it's not a big deal. I'm sure everyone can follow this. The amyloid protein that we call beta amyloid is actually a cleavage product of a much larger protein called the amyloid precursor protein, which I'm showing you here. And it's cleaved by these two enzymes, beta secretase and gamma secretase. And when that happens, this A beta molecule breaks off from the parent precursor and forms the plaque in the form of these particular species of amyloid called A beta 40 and 42. And we think that these plaques are related to the formation of tau and neurofibrillary tangles. And I'll show you how some of the evidence for that in a moment. So this is very basic, but it's very important to understand the relationship between the genetics of Alzheimer's disease and the clinical features. So it turns out that the genes that I mentioned that cause autosomal dominant Alzheimer's disease occur in several families. Occur in several locations on the genome. And one of the places they occur is mutations in this precursor protein itself. So very interestingly, the gene that codes for this amyloid precursor protein, if it carries the wrong mutation, will cause Alzheimer's disease. There are also two other groups of mutations that occur in this gamma secretase protein, which also cause Alzheimer's disease. And so all three of these kinds of autosomal dominant Alzheimer's disease, two mutations in the gamma secretase and one set of mutations in the amyloid precursor protein, all increase the amount of amyloid in the brain. They all increase the amount of A beta. And then, as it turns out, the ApoE4 allele is also associated with more brain A beta. So every form of genetic Alzheimer's disease is associated with increased production of A beta, which has led to this amyloid hypothesis of Alzheimer's disease. One of the best tests of this amyloid hypothesis came from a study that is called the Dominantly Inherited Alzheimer's Disease Network Study, DIAN. I'm not involved in this at all. And what they did in this study is they collected families who carried these abnormal, dominantly inherited gene mutations. So the people in these families either had Alzheimer's disease or carried the mutation and were at risk of Alzheimer's disease or didn't carry the mutation. And one of the things that interesting about these families is that the age of onset in the families tends to be preserved across generations. What that means is if you had a parent who had a gene mutation and got Alzheimer's disease at age 50, the chances are good that if you have a mutation, you'd get Alzheimer's disease within a few years of age 50. It's obviously horrible, but it is an excellent model for research because you can study people who carry the mutations who don't have symptoms and estimate how many years it will be until they get sick. And so that's exactly what these investigators did. And what they found, and this is a fairly complicated graph of multiple biomarkers. What they actually found is that the first abnormality in any of these biomarkers, amyloid deposition, tau, glucose metabolism, brain volume, and then this CDR, which is a cognitive test, the first thing that became abnormal was elevated amyloid in the brain. And this elevated amyloid started to appear about 20 years before individuals were expected to get sick. And so this was a strong piece of evidence that supported the amyloid hypothesis and linked the idea that amyloid deposition is the initial event in Alzheimer's disease. So let me show you some images and some data and show you why it's a little more confusing than that. So this is a PET scan using this PIB that we did in my lab. This is a patient with Alzheimer's disease who was, we scanned, who came from UCSF. And these bright red colors indicate more retention of the PET tracer. And so these bright red colors indicate a substantial amount of amyloid in the brain, just what we'd expect in someone with a diagnosis of Alzheimer's disease. This is an individual who's amyloid negative. These background colors of green and blue really don't indicate much of significance. And here's a normal older individual. Now we're following a cohort of cognitively normal older people in my lab. We've got about 200 people now that we've been following. This is an individual from that cohort, and this person has a lot of amyloid in their brain. And really, it's pretty much as this Alzheimer patient. And in fact, I didn't cherry pick this particular data. This is the data from the lab. And each of these data points is an individual. This is age. We always like to study a small group of people in their 20s because we don't think they should have any amyloid or any tau in their brain at all. And they have this value for amyloid here, which is, again, the PIB scan of 1, which means no amyloid in their brain. These are purple. These are our controls. These are most of our older controls. Also look like the young people. But then we have a handful of older controls whose amyloid levels overlap substantially with these blue dots, which are the Alzheimer patients. And in most studies, about 30% of cognitively normal people in their 70s and above have substantial amyloid in their brains. So right away, if amyloid is the cause of Alzheimer's disease, you're probably asking yourself, how can people be cognitively normal and have all this amyloid in the brain? And so that's just this little figure that I'm going to show you. The relationship between amyloid and cognition is weak. But I left all this black space for a reason, because other things are happening. And that's what's happening during the development of Alzheimer's disease. This amyloid is doing something to tau, and this tau is involved in neurodegeneration, and these processes together are driving the cognitive decline. So I'm just going to show you very limited evidence for this model. This is the amyloid hypothesis, basically, in a testable model. And I'm going to show you a small snapshot of data that supports this. So let's look at the relationship between amyloid and tau. This is one of the first studies that we did looking at this relationship. And what it shows you here is the effect of age on the deposition of tau. And what it's showing you is that as people get older, there's more tau accumulating in these brain regions that are colored. These are, again, the medial temporal lobes of the brain. And this is, again, the memory system of the brain. And this isn't really novel. We expected to find this. We know from autopsy studies that as people get older, there's more tau in these brain regions. This is the relationship between amyloid and tau. And what it's showing you is that as amyloid is going up, the tau is spreading. Now, these are cross-sectional, so we don't have longitudinal spreading measures here, but we subsequently got those. But here what you're seeing is that as the amyloid levels are higher, people have more tau throughout the rest of their brain. The surfaces of the brain, areas of the brain we call neocortex, in the temporal lobe, the parietal lobe, and the frontal lobe. So this was some of the first evidence to suggest that amyloid is driving tau from these initial brain regions into these other brain regions, widely dispersed throughout the cortex. So more recently, we've been able to look at some of these relationships. This is a study in patients with Alzheimer's disease. Again, we did this in collaboration with our colleagues at UCSF, where we measured the baseline biomarker, either PIB for amyloid, fluortaucipir for tau, and cortical thickness with an MRI scan, and looked at the rate of longitudinal atrophy. So the higher this number, the faster the brain is shrinking. And you can see that the more amyloid there was in the brain, the faster it shrank, but the relationship was relatively weak. The thinner the brain was at baseline, the faster it shrank, but also the relationship was relatively weak. But the more tau there was in the brain, the faster it shrank. And this was actually a very strong relationship. And so from this and a number of other studies in many labs now around the world, we're concluding that tau is very predictive of how fast the brain is going to shrink over time, much more so than the amyloid is. And then finally, there's a wealth of data, again, only showing you a snapshot. of the relationship between tau and cognition. This is the tau measurement in our older cognitively normal cohort. And what this simply is a correlation between tau in the brain and memory loss. And what this shows you is that the more people declined over time in their memory, the more tau they had in these brain regions. So tau is driving memory loss as well. So, you know, I think I said I was going to tell you what I think Alzheimer's disease is, and here's what I think Alzheimer's disease is. I think Alzheimer's disease is this medial temporal lobe tau that we all get as we get older. And in most people, this doesn't seem to have really terrible consequences. It might be associated with some mild memory loss that really doesn't affect a person's ability to function. But then some people, beta amyloid becomes deposited, and there's some interaction between this tau and amyloid, and we don't understand this. We don't understand this from research in my lab, although we have some thoughts. Molecular biologists and cell biologists don't understand it either. But somehow this interaction between amyloid and tau makes the tau very malignant, and it spreads throughout the brain and involves all these areas that are involved in not just memory, but language, visual ability, judgment, planning, et cetera. And this is the beginning of Alzheimer's disease. And this in turn leads to shrinkage of the brain and neurodegeneration. So now I'm going to switch to the idea of therapy. And there's been a lot of different kinds of therapies for Alzheimer's disease. I'm going to talk about two approaches. The first one is the one that's gotten all the headlines, and they've generally been depressing headlines. And that's immunotherapy. And what immunotherapy is targeted antibodies to the amyloid or A-beta molecule. These immunotherapies are, up until recently, have been infused intravenously. So they're very expensive, very difficult to administer. More recently, there's some evidence that subcutaneous administration might work. But up until recently, they've all been administered, requiring administrations every 6 weeks or so. And one of the things we've learned that anti-amyloid therapy is effective at lowering amyloid. So these were the first studies, this was the first big study that looked at immunotherapy for Alzheimer's disease. And what it showed is in the first study, in the E4 carriers, this is the placebo group, this is the drug group. And what you can see is that the placebo group's amyloid went up, and the treated group's amyloid was stable. In the E4 non-carriers, it was really difficult to see a signal. So it was a weak effect, but it was encouraging to people. And the one thing that you can say for sure is that over time, these drugs have gotten better and better at lowering amyloid. And this is the most recent study of a drug called donanemab. This bapineuzumab was the first study. This is a phase 3 study. It was considered a failure because patients didn't clinically improve. This was a most recent study. It was a phase 2 FDA study. It was highly successful at lowering amyloid. And in fact, almost over two thirds of the treated group actually converted their scans to amyloid negative, removed all the amyloid from the brain. And in this phase 2 study, there was actually a clinical signal that people had improved. Not enough to support an FDA application, but it was a cause for optimism in the community interested in these kinds of therapies. So this is really interesting, but I think you learn a lot from spending a minute talking about this interesting study that probably you've all heard about, a drug called aducanumab, marketed as Aduhelm. Even before the FDA released their judgment on this drug, people were arguing about it. And this essay in the New York Times pointed out why the clinical data were so weak. The FDA nevertheless went on to approve it in June. This created a ruckus. A lot of people were very upset. And in fact, 3 of the advisors who were on the review committee for this drug quit. And you can see this is a drug that had a marginal or questionable benefit and was going to cost $56,000 a year for treatment. So what actually happened in this study? What's the reality? So the first weird thing about this study is that the Data Monitoring Committee, the Data Safety Monitoring Board, looked at the data in 2019 and decided there was no way that the drug was going to be effective. So the trial was halted before it was concluded because of futility. But then when they went back to look at the data months later, there were two simultaneous clinical trials, and they decided that one of these trials was actually positive. It met its primary endpoint. So the company took that one out of two trials to the FDA, and the FDA convened an advisory panel. This is the one I mentioned, where 10 of the members of the committee voted against approval. One was uncertain. No one voted in favor of approval, but the FDA went on to approve it. And they approved it based on what they call an accelerated pathway, which basically used the surrogate endpoint that the drug lowered amyloid in the brain in the PET scans. So this is an interesting pathway. It was really developed during the HIV era to use surrogate endpoints so that drug development could be speeded. And it was very effective in developing good treatments for HIV. But there was a lot of argument about approving a drug on a surrogate endpoint because there wasn't good evidence that lowering amyloid made anyone better. So this was also highly controversial. And the next step was that it went to Center for Medicare, which made a national coverage determination that requires what's called coverage with evidence development, which means that they will approve this class of drugs, but only if individuals who are put on these drugs are either enrolled in a subsequent clinical trial or a national registry. And so that's where this entire class of drugs stands right now in terms of their approval. And we can come back to this if you want to talk about it. It's a very interesting kind of coverage determination. So just to show you the data from aducanumab, these are the PET scans showing you the baseline and the placebo follow-up, but then the different doses, clearly removing amyloid from the brain. And these are the two clinical studies. One was called Emerge, the other called Engage. They were large studies with 1,500 participants in each of them. And you can see there was a dose effect on lowering the amyloid in the brain. So again, very effective amyloid lowering, but not so great clinically. Here's the positive study, the Emerge study. This is the placebo group. Higher is worse. This is the high dose treatment. And this was actually a very small effect and greatly argued as to whether it had any clinical benefit or not. And here's the other study, the Engage, that didn't meet its endpoint. So that's the actual data. I just want to point out that, as I said, these trials require intravenous infusions every 6 weeks. They're associated with what you're seeing here is swelling of the brain or cerebral edema, which requires regular monitoring with MRI. This is usually asymptomatic, but not always. It's kind of scary looking. I certainly wouldn't want it in my brain. And it's very expensive and it happened in the aducanumab trial in 25 to 35% of participants. So this is a big problem. So there've been a number of amyloid immunotherapy trials that have failed. And so the question is why? So one reason that I'm going to show you is that as people have looked more and more, and in fact, we've known this for decades, the neuropathology of patients with Alzheimer's disease is more complex than just plaques and tangles. There's vascular disease. So I mentioned that stroke and vascular risk factors are associated with an increased risk of Alzheimer's disease. And that's probably because it potentiates the effects of the amyloid and tau proteins. And there are other proteins that are deposited in the brains of older people as well that can cause cognitive decline and dementia. So Alzheimer's disease is rarely an isolated finding in the brain. There are individual factors such as particular resilience or vulnerability that are important. And the incubation period for Alzheimer's disease is very, very long. And treatment is too late. Once people have symptoms, it seems very unlikely that effective therapies are going to have a big effect. So just quickly to show you some of this pathology data, this is a pie chart of the pathology that's seen in the brain of people with a diagnosis of clinical Alzheimer's disease. This little sliver here are the cases that have just Alzheimer's disease. These are cases that have Alzheimer's disease and vascular pathology. These are cases that have Alzheimer's disease, vascular pathology, and other brain degenerations like the degeneration that's associated with Parkinson's disease, for example. So I'm going to zip through this because I don't want to belabor this point. I just want to clearly make the point that when clinicians diagnose Alzheimer's disease, yeah, there's usually Alzheimer's disease in the brain, but there's almost always other pathology that coexists with it. And whether an amyloid-lowering treatment is going to make someone better or not is probably going to depend on how much of this other pathology there is and what kind of effect it's having on their cognition. I'm going to skip this as well because I want to talk about some of all that. And then there's the issue of resilience. So these are two individuals. One of them is cognitively normal. The other has mild cognitive impairment. And these are their amyloid scans. Now, you're going to have to take my word on this because I just know the data, but they have pretty much the same amount of amyloid in the brain. But this person has mild cognitive impairment, and this person is normal. If you look at their tau PET scans, they're different. And this person, for the same amount of amyloid, has a lot more of this tau in the brain. And this person also, because probably they have more tau, they have a lot more shrinkage of the brain, a lot more atrophy, a lot more neurodegeneration. And what the factors are that are making this person more resistant to the pathology or resilient to the pathology versus this person who's more vulnerable to the pathology, we really don't understand. I just want to end this section with a recent study. And we're really entering a time now where in the next several months, there are going to be a lot of big studies that are reading out with drugs that are very, very effective at lowering beta amyloid. And the one that came out most recently, the only information I have is a press release. This is a drug called lecanemab. It is a very potent amyloid-lowering immunotherapy. And in this study, the primary endpoint, which was the same as aducanumab, was met. And it's a reduction of 27% versus placebo at 14 months, and the raw score of a 0.545 difference. It also hit most of its secondary endpoints. So the company is overjoyed about this. It's Biogen and Eisai. It's undoubtedly going to be approved by the FDA. And you might ask, how clinically significant is it? Well, it's a little hard to say, but this difference in score could mean the difference between being cognitively normal and having mild cognitive impairment. So this drug, lecanemab, may well be the first approved drug for Alzheimer's disease that actually modifies the disease. So just going to end with a couple of other points about other approaches, another approach to treatment of Alzheimer's disease. And that gets back to this issue of risk factors for Alzheimer's disease. So the study that really opened the door to this was a study in Finland called the FINGER study, where they picked an interesting group of individuals who are at risk for cognitive decline based on a high body mass index, being older, having high blood pressure, high cholesterol, physical inactivity, and slightly lower than expected cognitive performance. And they had what's called a multi-domain lifestyle intervention. So they had dietary advice, physical exercise, cognitive training, blood pressure control, socialization. This wasn't just advice. This was regular meetings with healthcare workers and guides, basically, who are involved in keeping people on the program throughout the 2-year randomized treatment. And at the end of this treatment, there are two interesting findings. First, higher is better. These are cognitive tests. Everyone got better. So the control is in red, and the control is in blue, and the intervention's in red. And you can see even the control intervention, which was just health advice, Even those folks got better, but the intervention, the individuals treated with the intervention improved substantially more. And so this opened the idea that why bother treating just blood pressure? Why not treat every lifestyle or vascular risk that we can? And so that spawned many similar trials that are now ongoing around the world. There's one in the United States. This is called the Pointer Study. And so this study is currently, it's centered at Wake Forest. It's enrolling 2,000 individuals, very similar to the ones I described in the finger study. These individuals either get as a control, a self-guided lifestyle intervention with education and support, or a structured intervention, which is happening at YMCAs 4 times a week, intensive one-on-one involvement, a Mediterranean diet, computerized cognitive training, group meetings, et cetera. And so this study is still recruiting, but I think will be very interesting to see if this can protect cognitive function, which is the outcome. And as part of this study, we're actually interested in getting back to this idea that maybe these kinds of interventions can actually affect the pathology in the brain. So we've appended this imaging study in Berkeley where we're actually looking at what's happening to the brains of individuals who are in this study. And these folks are going to get MRI scans and PET scans to measure amyloid and tau to see whether imaging measurements will improve with the intervention and whether they can identify people who are going to benefit. So going back to this model of Alzheimer's disease, the simple idea is this amyloid plaque leads to some change in the tau, which drives brain atrophy and neurodegeneration and cognitive decline and dementia. But we also know there are other degenerative disorders, protein aggregates, and also cerebrovascular disease that contribute to this end stage of the process where the brain degenerates. And so these things are going to have to be treated probably as well. That's not to say that anti-amyloid therapy may be useless. It's not clear how useful it's going to be in any given individual, because that's going to depend on how active all of these other processes are. There's also the possibility of anti-tau therapy, which is definitely in trials using immunotherapy as well. There's a whole idea of vascular risk factors and lifestyle intervention. And then there's so many other targets for therapy that are being looked at because the molecular mechanisms that underlie these changes are getting understood a little better all the time, but we still don't have a good handle on them. So we clearly need other approaches as well. So that's the end of my talk. I think I left some time for questions and be glad to entertain them. Thanks.
Great, thank you so much for that. Really great presentation. I learned so much. I'm sure there will be questions. Let me see if there are any in the chat, and people can feel free to raise your hand or unmute. And let me see, I think I see one thing in the chat. Just wondering if anyone is aware of any studies tracking, oh, okay, that's for a different one. Okay, anybody have a question they'd like to raise?
Mindfulness in Alzheimer's, yeah. So there is an ongoing study in Europe called the Meditaging Project, clever name, on meditation and cognition. That study hasn't read out yet. It's a big study in Europe. The EU funded it, and it's still ongoing, but it's a randomized trial actually of meditation. Yeah.
Great, thanks. Sally has a question. Are the lifestyle interventions expected to be able to help people with the gene mutations that deterministically cause or related to early onset? Yeah,
that's a good question. I think not. I mean, I wouldn't think so. Let me put it that way. I think, first of all, if you think about who's in these studies, right? These are folks who are sort of at increased risk, but definitely not showing, frank disease. So first of all, you'd have to ask the question is, how beneficial is this going to be in people who already are sick? And my guess is that any disorder is going to be, any intervention is going to be less effective in people who are sick. My concern about these autosomal dominant mutations is that they're so deterministic that virtually nothing will affect them. And it's unfortunate. But again, if you look across people who carry these gene mutations, there's some evidence that ages of onset are variable, within families, but as I suggested, not that variable. So I don't think environmental factors are going to play a huge role in modifying these autosomal dominant mutations. There's a question here about care homes and an increase in progression of the disease. Interesting question. I don't know the answer to that. I think, again, I guess it probably depends on why people are in care homes. One of the things that's important to understand about this disease is that once it's manifest, the destruction in the brain is so severe, and the brain has such little capacity for self-repair that I think interventions are not going to be super helpful. So I think the idea that, so if someone is in a assisted living or SNF or something like that because they have Alzheimer's disease, I'm not sure, again, that these interventions are going to be that effective.
There's a question on, is there any studies on psychedelics and Alzheimer's?
Very interesting. No. So we're interested in doing one of these studies. I have a student in my lab who's interested in looking at the effects of psychedelics on aging. So I don't think we're going to be giving psychedelics to people with Alzheimer's disease anytime soon. But I do think it's possible that we'll be giving them to older people to see how they affect a variety of different kinds of cognitive processes. So I think that's an interesting question. We're Berkeley's in the vanguard on this, and I hope we can get involved in it.
Great, thank you. Is there any similarity between long COVID and Alzheimer's disease, or any evidence that the root cause is CNS inflammation?
Yeah. Oh, yeah. Great question. So I'm not aware of any relationships between COVID of any sort and Alzheimer's disease. I'm sure someone with Alzheimer's disease who gets COVID might well deteriorate. It's not cognitively, wouldn't be surprising. But there is definitely a very important story on inflammation in Alzheimer's disease, which I entirely left out of my talk just because of time. And I think there's no doubt that there's a role that inflammation plays in Alzheimer's disease. And one of the reasons I left it out is because it's not really clear what role inflammation does play. It may be sort of a bimodal role where at one point in the disease, it's actually harmful, and at another point, it may be beneficial. But we just don't understand whether the effect of whether inflammation is beneficial or one of the initiating factors in the disease. But it's undoubtedly plays a role. Treatment with anti-inflammatory drugs has been tried, and so far has been ineffective.
Thank you. Is the surrogate endpoint that you mentioned used by the FDA in clinical trial approval potentially more useful for diseases which have a clearly defined necessary cause like HIV than for Alzheimer's?
Well, that's the big question, right? That's what a surrogate marker is supposed to be. In fact, is a marker that I forget the FDA wording is lowering the surrogate is reasonably likely to improve the clinical outcome, right? So for HIV, that reasonably likely, I think, has been pretty well established. But the problem is for Alzheimer's disease, many would argue that it hasn't. So I guess, I think where this field is heading is that surrogate markers are not going to do the trick in Alzheimer's disease. No one is going to be, well, I shouldn't say no one, but I think the field is not going to be moved by amyloid lowering without a clinical response. And I think once there is good evidence that really profound amyloid lowering will produce a clinical response, then maybe we can move into the surrogacy phase, right? This is true for all you know, for all kinds of treatments. We routinely use surrogates, you know, markers for lowering blood pressure or cholesterol, right? But now we know there's a strong association between blood pressure and cholesterol and health outcomes. So we have to establish those much better before we can move to surrogacy. And once that's established, then maybe clinical trials will get easier.
Great. Well, thank you so much. We're at time, but really appreciate everybody joining and appreciate you sharing your work with us, Bill. So thank you, everybody, and have a good day. Yeah, thanks so much for sharing your excellent work. Bye, everyone. Bye.
Bravo.
Yay.